We've gotten to this part of the respiratory system where we learned about the overall structure of human lungs, that the right side has got three lobes, the left side has only two lobes. Um, and we learned about the structure of the carina. Before we leave the point of the carina, I wanted to give you a view of what it would look like if you actually took one of those flexible fiber optic endoscopes and you looked down a patient's trachea. If you look down a patient's trachea, this is basically what you would see. You would get right here to this area called the carina, and there would be the right primary bronchus and the left primary bronchus. And one of the things I'd like you to notice is that when you get here, it is a straight shot going down the right primary bronchus. See how you can see right down it? Whereas the left primary bronchus, you kind of need to take a left turn to go down the left primary bronchus. Why is that interesting? The reason that that is interesting is that there's actually more asymmetry to the structure of the bronchial tree of humans than this particular image would indicate. Actually, the right primary bronchus goes a little more straight down than this, and the right goes a little more straight down than this, and the left goes off at a little more of an angle. And why is that interesting? Because if a kid has inhaled something they shouldn't have, like Barbie's shoe, then Barbie's shoe is going to get right to this part right here and keep on heading down the right primary bronchus. Things that have gotten inhaled, whether that's Barbie's shoe or vomit from a patient that's very sick, is very likely to go down into the right lower lung lobe because of the asymmetry of the bronchial tree in humans. Now, when we get past all of the bronchi, we will get to structures that are called bronchioles. Now, I know I am guilty of this too, that I usually think of venules as just being really small veins and arterioles as really small arteries. I got to admit, I do. I just want to make a point that bronchioles are not just really small bronchi, right? Bronchioles are important because they do not have any cartilage propping them open. And that ends up being very important, particularly for a health problem called asthma that we'll talk about in a moment. So remember, bronchioles are different from even the smallest of the bronchi in that bronchioles do not have cartilage in them. So here is a very, very tiny bronchiole. There's no cartilage in the wall at all. Now the bronchioles, they end in alveolar sacs, and the alveolar sacs are groups of alveoli. So this is an alveolar sac, this whole area here, and each one of these little guys would be an individual alveolus. And so the individual alveoli are kind of like rooms that all open onto the same atrium. And the whole set of alveoli and the atrium are called the alveolar sac, and they get attached to a bronchiole, and that's the structure of your lungs. Here's something that always blows my mind when I look at it. This is an image looking through the microscope at some lung tissue. And I want you to notice that everywhere that it is white, the only thing that belongs there is air. And that means that the amount of actual human cells, human tissue that is present in this lung, that's a lot less than a quarter of all of that space that is being occupied there. Your lungs are much more air than they are you, right? And inside of that little tiny line is actually just 4 layers of epithelial cells, 4 layers of squamous epithelial cell, plus a little capillary blood vessel going by. We'll look at that more closely. Before we do, let's take one more look at what's called the bronchial tree, all right? So this isn't perfect, but remember your little alveoli, the little air sacs, they're going to be leaves, and the leaves are attached to bronchioles. The bronchioles would be like the little twigs that are attaching the leaves to the tree. And then we've got different sizes of branches and ultimately a really strong trunk. Now, I want you to imagine that I have got a tree in my front yard, and the tree in my front yard has got a tumor right there in the middle of that big main branch. Don't ask me how the tree got a tumor. It just did, right? Now, if I have the tree trimmer guy come out and I say, hey, tree trimmer guy, I want you to remove the tumor from my tree. And the tree trimmer guy is going to go, yes, ma'am, I'll take off that branch. And I say, oh, no, no, no. I don't want you taking off the branch. Leave the branch. I just want you to take out the tumor. Well, the guy would think I was pretty stupid, right? Because he knows that if he takes out this little part of the trunk, that there's not going to be any water and stuff getting to those leaves and that the tree branch would end up dying. He knows that. So do you. What does that have to do with lungs? Well, lungs are built the same way. You know, in our anatomy class, we've more been thinking about lungs from the point of view of these tubes, and I guess that's natural. What is not clear to you from our anatomy lab is running on either side of these tubes, like right there, and right there, and right there, and right there. Those are going to be the pulmonary arteries, which we now know are blue, and the pulmonary veins, which we now know are red. So this would be like one of those big branches on my tree. If there is a tumor here, I can't just cut it out. Because if I just cut it out, I won't just removing the tumor. There will be no way for air to get from here to there. There'd be no way for blood to get from here to there or from here to there. So when it comes to tumors developing in the lungs, tumors that develop in the lungs generally need to be removed by what's called a wedge resection. This particular image has been colorized to show you the different lobules inside the lungs. So if I had a tumor right there, I couldn't remove just that tumor. I'd have to remove all of this dark blue stuff, right? If I had a tumor right there, I'd have to remove all of that red stuff. I can't just remove a little chunk there. And that's how removing tumors from lungs is so different from removing them from your liver or something like that. But worse than that, when people have got lung tumors that are caused by cigarette smoking, lung tumors caused by cigarette smoking tend to happen here in the primary bronchi. So if I've got a great big lung tumor here in the primary bronchus from cigarette smoking, I would have to remove an entire lung from that patient. And that is very difficult to accomplish and maintain a good quality of life for the patient after you've removed it. That's one of the reasons why, it's not the only reason, but it's one of the reasons why we don't generally remove those tumors with surgery. We try to treat them with different chemotherapy agents. All right, we have talked about things that have influenced blood pressure and stuff. Let's talk about factors that affect resistance to the flow of air through the lungs. The factors that affect the resistance of airflow are basically two, okay? The first is pulmonary compliance. Pulmonary compliance is a measure of the stretchiness of lung tissue. Lung tissue is meant to have a lot of that protein elastin in the structure of the lung. And that makes it so that it's very easy to inflate the lung, but when you remove the pressure, the lung empties itself really good. When you've got young lungs like you do, then it's really easy for you to inhale. And then when you just relax your muscles, your lungs empty themselves, right? Compliance means that for a person with healthy lungs to exhale, all they have to do is relax. The lungs are like a really brand new stretchy balloon, and when you let go, they empty themselves. That factor is pulmonary compliance. Pulmonary compliance is decreased by a few factors. Cigarette smoking definitely damages the structure of lungs. And there are disease conditions like sarcoidosis or lupus that also can replace the elastin with collagen. Elastin is stretchy, collagen is strong. So if you get rid of stretchy and you put more tough, it's harder to inhale. By the way, those of us who live here in the LA basin for very long, the air pollution around us is also damaging our pulmonary compliance. Let's talk a little bit about the second important factor that changes resistance to airflow in our lungs. That is the diameter of our bronchioles. The diameters, the radii, whichever, okay? How big are our bronchioles? Now, I said bronchiolar diameter. It is true that all of your airways, that there are factors that can make them relax and get bigger or contract and get smaller, right? But for all of those other airways, when the smooth muscle around that tube constricts, it can only constrict it a little bit because there's a piece of cartilage, right? If this is relaxed, my cartilage, my hands, and then I cause constriction, when the two ends of the cartilage touch, I just can't make the tube smaller than that. But remember what I just told you, that the bronchioles do not have cartilage in them. So the bronchioles, if you ask them to constrict, they can just go super, super, super tiny. One of the things that causes a problem called an asthma attack is bronchiolar constriction. Bronchoconstriction, when I have asthma, I had wicked asthma when I was a kid. And when I heard that there was smooth muscle wrapped around all of my airways, and that when it constricts, that it makes me have an asthma attack, I thought, well, who's the brain surgeon who wrapped smooth muscle around my airways? Because from my point of view, I don't ever need my airways to be smaller. So why is that stuff there? Well, the reason there's muscle wrapped around our airways is so that your body can modulate the relative benefits of big open airways that are better at getting lots of air into your lungs fast with the need to clean and warm and humidify the air, right? Remember, there are two things going on. We need enough air to keep ourselves going, but we need that air to be very, very clean when it gets there. When you are relaxed and not doing very much, just hanging out, right? Your airways, they constrict quite a bit. And you don't notice it because you're not having trouble breathing. You're not exercising, right? When you're just hanging out there, it's fine if our airways are only this big. As long as our airways are only that big while we're sitting around the fire or whatever, that means that the air is going to be cleaner and warmer and more humid when it arrives in your alveoli, right? But there are times when you need your airways to be really big and open. Like if you're running from a bear. If you're running from a bear, you're like, open up my airways, baby. I'll worry about dirty lungs next week, right? If I see next week. So while we're in rest and digest mode, everything's copacetic, our airways are designed to be smaller. But if we're terrified by something and we need all of that air, then adrenaline, which is epinephrine, goes into our body. It causes those to relax. And then big gulps of air can go in and out real easily. And we'll worry about dirty lungs next week, right? So bronchodilation is caused by the sympathetic nervous system, that's the fight or flight part, and by adrenaline. The medical word now for adrenaline is epinephrine. Bronchoconstriction is normally going to be around as long as you're not terrified, but not enough to cause an asthma attack. Asthma attacks happen when your body's immune system makes a mistake and thinks that there's some dangerous reason to not allow much air in. And that is going to cause asthma attacks. We're going to start with a little diversion on the effects of asthma at the beginning of our next lecture.